and welcome to the nervous system. This is segment 4 of 5, and in this recording I will cover slides 57 through 114. And in this recording we'll pick up at topic number 4 here, functions of the spinal cord, and we'll go through topic number 5, select functions of major brain regions. And then the last two topics will be covered in the final segment. And in this recording, we will talk about most of these phenomena. All right, functions of the spinal cord. You know that the spinal cord is part of the central nervous system, but what does it do? The spinal cord is continuous with the brainstem, and it emerges from the cranial vault via the foramen magnum, which really means big hole. And it's that large opening in the occipital bone at the base of the back of the skull. The spinal column, or the spinal cord, I should say, is encased in bone. It passes through the vertebral canal, which is a canal-like structure created by the individual vertebrae as they stack on top of each other. The spinal cord terminates around lumbar vertebra 1, 2. It's different in different bodies, but the spinal cord ends as a pointed tip called the conus medullaris. And emerging from the conus medullaris is a cluster of spinal nerves known as the cauda equina, or horse's tail. And those spinal nerves continue to descend through the vertebral canal and into the sacral canal. Each segment of the spinal cord, where a segment of the spinal cord is defined by the particular vertebra that it's passing through. And you can see the names of each vertebra there. We identify the vertebra by the region of the vertebral column: cervical, thoracic, lumbar, or sacral. And then each vertebra is given a number. There is a mistake. Oh, I see. I thought there was a mistake in this image, but it's pointing out the pair of spinal nerves. Okay, so for each segment of the spinal cord, there is a pair of spinal nerves that emerges or connects to the cord, depending on your perspective, between the neighboring vertebrae. And also in this image, we can see some nerve plexuses. So starting at the top on the left, we can see the cervical plexus, and then the brachial plexus. There's a lumbar plexus and a sacral plexus. And so a plexus is a braided network of nerves. So now we're looking at a smaller section of the spinal cord, and the vertebral column has been removed. We can see that there are 1, 2, 3, 4, 5 spinal cord segments here, because we can count 5 pairs of spinal nerves that connect with the spinal cord. We can see the spinal cord is covered, surrounded by the spinal meninges, and the spinal meninges are continuous with the cranial meninges, and they have the same structure. The meninges have 3 layers. Going from superficial to deep, the exterior or most superficial layer is called the dura mater, and that's a tough fibrous coating. And then deep to that is the arachnoid mater, arachnoid because it has a web-like sort of gossamer structure. And then deep to that is the pia mater. The pia mater is very thin, and it clings to the exterior surface of the central nervous system. Blood vessels also run through the pia mater. Looking at the cord itself, we can see, now I'm at the top of the image, we can see that there is both gray matter and white matter in the spinal cord. This is also true of the brain. Gray matter in the central nervous system consists largely of unmyelinated neurons, whereas white matter consists largely of myelinated neurons. In the spinal cord, we see that the gray matter is deep and the white matter is superficial. And we'll learn about the difference in function between the gray matter of the spinal cord and the white matter of the spinal cord in just a few moments. An important landmark on the spinal cord is the anterior median fissure. And you can see that label near the top right of this diagram. The word anterior means toward the front of the body, and median means middle. So this is a deep crevice that runs along the anterior aspect of the spinal cord. And it's a landmark that can help you get your bearings. What's front, what's back, what's anterior, what's posterior. Here we're looking at just a single segment of the spinal cord. We can see one pair of spinal nerves, and spinal nerves are called mixed nerves because they contain axons of sensory neurons as well as axons of motor neurons. So sensory neurons carry afferent signals toward the central nervous system, and in this diagram the sensory neuron is indicated in blue, like a turquoisey blue. And motor neurons carry efferent or outgoing information away from the central nervous system to effectors, where effectors are muscle cells or gland cells. We can see how this spinal nerve physically connects to the spinal cord. There are nerve roots, and if we look at where those nerve roots join the spinal cord, we can see that the axons of neurons, sensory neurons and motor neurons, emerge with the nerve or emerge as part of a nerve root coming from the gray matter of the spinal cord. Notice that as we're looking at that flow of incoming sensory information and outgoing motor information, notice that here we don't see any involvement of the white matter. This is a section of spinal cord viewed through a light microscope. It has been stained. We can see distinctly the gray matter and the white matter. We can also distinctly see the anterior median fissure. Let's start with the gray matter. The gray matter of the spinal cord includes neurons or portions of neurons that are responsible for delivering information into the spinal cord or for giving rise to information that exits the spinal cord. So sensory information travels toward the central nervous system. Those afferent signals enter the gray matter of the spinal cord via a distinct horn called the posterior gray horn. So where you see that sensory neuron here, let me pick a color, let's try green because there doesn't seem to be a lot of green. Where you see this blue sensory neuron, this blue sensory neuron is delivering sensory information into the spinal cord via the posterior gray horn. So this little blip of gray matter right here is the posterior gray horn. Notice this structure here. This is a posterior root ganglion, also known as a dorsal root ganglion. And notice that the cell body of that sensory neuron is in the posterior root ganglion, and then its axon continues via the posterior gray horn into the gray matter of the spinal cord. Motor information exits the spinal cord through other gray horns. This distinct bump right here, this is called the lateral gray horn of the spinal cord. And in the lateral gray horn, that's where we find the cell bodies of motor neurons that are part of your autonomic nervous system. And in the anterior gray horn here, this is where we find the cell bodies of motor neurons that serve your somatic nervous system. So in blue, we have a sensory neuron. In sort of a greener color, we have a motor neuron that's serving the autonomic nervous system, and in red, we have a motor neuron that is going to deliver information to skeletal muscle as part of the somatic nervous system. And then we have an interneuron in purple, and I hope you remember that interneurons are neither motor nor sensory. And in this particular diagram, this particular interneuron is connecting, is allowing information transmitted by that sensory neuron to be passed on or not passed on, depending onto that red motor neuron. What's not particularly highlighted in this diagram are the functions of the white matter. In the white matter of the spinal cord, information can ascend or descend through various levels of the spinal cord or between the brainstem and the spinal cord. So information travels up and down in white matter tracks, which I'll show you in just a little bit. And information gets on and off the spinal cord through pathways in the gray matter and the spinal nerve roots and the spinal nerve. A useful analogy for understanding how information travels through the spinal cord is to think about an elevator in a very tall building. And this tall building has many floors, just like your spinal cord has many segments. And you can ride the elevator to pass between segments of the spinal cord, or you can ride the elevator to get from one floor to the next floor, from let's say floor 17 down to floor number 4. And if you want to get off on a particular floor, you'll have to exit the elevator. So the elevator shaft is analogous to the white matter tracks in the spinal cord. And then the doors, where you get on and off the elevator, that's analogous to the gray matter that connects to spinal nerves. This image highlights white matter tracks of the spinal cord. Tracks are identified as either being ascending or descending. To ascend means to go up, and to descend means to go down. So sensory information that has entered the spinal cord will travel up to other levels of the spinal cord or will travel up into the brain stem. And motor information will travel down. That motor information maybe it started at some superior spinal cord level and it descends to a lower spinal cord segment where that outgoing motor information might exit the spinal cord through motor neurons. Spinal cord injuries are devastating, life-threatening, as you're probably familiar with. There are 3 basic types of spinal cord injuries: a transection, a hemisection, and a compression. Trans means across. So in a spinal cord transection, the entire cord is sliced or broken. And the slicing usually occurs from a piece of bone that severs the spinal cord completely. And then a hemisection, hemi means half. A hemisection means just a portion of the spinal cord is sliced, but the slice doesn't completely transect it. And then there's a compression. Something is applying pressure onto the spinal cord. And so which injury does this most resemble? This is a compression injury. And in all 3 types of injury, the major problem is communication between levels of the spinal cord is disrupted. So look at the injury that we see on this image. Information can neither travel up the spinal cord past that level of injury, and information cannot travel down the spinal cord beyond that level of injury. So communication between the upper portion and the lower portion is completely cut off. No motor information and no sensory information can pass through the site of injury. Here are some of the common causes of the 3 types of spinal cord injury. Transection and hemisection typically result from some type of trauma, a fall, a car accident, whereas compression can result from either trauma or disease. So a car accident, a fall, could result in a fractured vertebra, which would then compress the spinal cord. But then we have other progressive disorders like a herniated disc, a tumor, or an infection. A herniated disc is caused when an intervertebral disc, these pads of fibrocartilage that occur in between the bodies of neighboring vertebrae, they have sort of a softer gel-like center. And when the exterior surface of the disc ruptures, that gel-like substance can bulge out and put pressure on the spinal cord or the spinal nerves. And a tumor is a collection of cells that shouldn't be there. Cell division has gone haywire, and this mass of cells is called a tumor, and it can put pressure on the spinal cord. And then infection, I think you're familiar with that term, a bacterial or a viral infection. Bacteria can get into the bone, can get into the vertebral canal where they eat and divide and eat and divide, and that's what bacteria do. And that can also bring about an immune response that leads to inflammation. And that inflammation, as well as the bacteria, can put pressure on the spinal cord. So once again, spinal cord injury stops the transmission of information. Information cannot pass the site of injury. And so the degree to which the body is affected depends on the level of the injury. So spinal cord injury results in numbness and paralysis because both sensory and motor information are disrupted. Here are 3 examples of spinal cord injury identified by the level of transection. So if this transection occurs high in the cervical region of the vertebral column, sensory and motor function will be affected from the neck down, and the person will require a ventilator. That motor output sent to the diaphragm to cause pulmonary ventilation, that motor output can no longer travel to the diaphragm, and so a ventilator is required to ensure that the person continues to breathe. If we're much lower, if we're in the thoracic region, between level 8 and level 9 of the thoracic region, the person will experience lack of sensory and motor function from the waist down. They may be incontinent. And if we are much farther down, if the spinal cord injury occurs at the inferior tip in the vicinity of the conus medullaris, the person will have a loss of sensory and motor function from the hips down. So they may have the ability to control their bladder and their bowel, but they won't have any sensory or motor function in their legs. The goal in treating a spinal cord injury is to restore function of the spinal cord, to allow sensory information to travel across the site of injury and to allow motor information to travel across the site of injury. So we need to induce neurons to grow through the site of injury and reestablish their synaptic connections with other cells. And stem cell therapies look particularly promising. You've probably been hearing about stem cell research for, depending on how many years you've been on the planet, but for a few decades now. And we are on the cusp of some amazing discoveries. Different research groups are taking different approaches using stem cells from different parts of the body. So mesenchymal stem cells come from the bone marrow. And pluripotent stem cells, we can take them out of the stratum basale and the epidermis. We can take your own skin cells. And then there are other sources of stem cells. But regardless of the source, we, the body of science, we can apply particular signaling molecules as well as electrical stimulation to induce these stem cells to become neurons And then we can surgically implant these stem cells at the site of injury and continue to encourage them to differentiate into functioning neurons and form synaptic connection with cells that are existing in the patient. And so far, we have not fully healed a transected spinal cord, but in some clinical trials, we have been able to achieve some improvement in sensory function, in motor function. In some patients, bladder and bowel function has been restored. This is a very young field, and it's not clear which techniques will emerge as the most successful or how successful they will be, but it is very promising at the moment. All right. Earlier, I described the human brain as being the most complex structure in the known universe. That still is true. And I hope someday you go on to take a neuroscience course. It's fascinating. And almost every physiologist I know has a deep interest in neuroscience. So this is not a neuroscience course, and I'm sad to have to limit the information to just select functions of major brain regions, but I've tried to choose particularly interesting things to present to you. So let's start with the major brain regions. There are 4 major brain regions, and I'll start with the brain stem. So the brain stem is continuous with the spinal cord, and the brain stem includes 3 subregions: the medulla oblongata, the pons, and the midbrain. Superior to that is a region of the brain known as the diencephalon, and the diencephalon includes several subregions. We will spend most of our time talking about the thalamus and the hypothalamus. And then superior, or sort of superficial to that, is really the cerebrum. And the cerebrum comes in 2 hemispheres. We're only looking at 1 hemisphere in this mid-sagittal section of a brain. And then our 4th major brain region is the cerebellum, and it's posterior to the brainstem and sort of inferior to the cerebrum. This is a human brain taken from a cadaver, where again, let me just review those 4 major brain regions. There's the brain stem. There's more centrally located the diencephalon. The big wrinkly parts on the outside, that's the cerebrum. And then in the back, near the base of the skull, is the cerebellum. Brain functions occur because neurons are connected to each other in networks. These are called neural networks. And it is not the case that individual brain functions occur from individual regions of the brain alone. There might be part of the brain that is in charge of a particular function, but that part of the brain is connected via neural networks to several other brain regions. And these neural networks work together to bring about particular functions of the brain. This is a super cool image. It was taken using a technique called diffusion spectrum imaging. So this is the brain of a monkey, a macaque, which is a model organism. And radio-labeled water is injected into the central nervous system, and then a series of images can track the movement of those labeled water molecules. You know, osmosis happens. And so as those labeled water molecules move along axons and from one neuron to the next, we can track the pathway of those water molecules, and then we can develop an artificially colorized image. It's not that the water molecules are actually blue, but we can follow the pathway of particular radio-labeled water molecules and then later on color that pathway blue. All right, here are those major brain regions again. We're looking at an artistic representation of an intact brain, so you can see which structures are superficial and which structures are deep. Here are select functions of particular regions of the brain stem. Some of these functions have come up earlier, some of them will come up in the future. So in the medulla oblongata, and if you look at the picture, that's the blue region of the brainstem, the inferior most region of the brainstem. In the medulla is where we find the cardiovascular center, and we did talk about the cardiovascular center as an integration center for a reflex arc, and we will continue to talk about that when we get to the cardiovascular system. Also located in the medulla are reflex centers for vomiting, coughing, and sneezing, and there's a reminder here that these are all autonomic cranial reflexes. They are governed by the autonomic division of your nervous system that concerns smooth muscle, cardiac muscle, and glands. And they're cranial reflexes because the site of integration is located in the brain rather than the spinal cord. And then if you include in your gaze now the pons, located in both the pons and the medulla are a series of nuclei, series of centers that are collectively referred to as the respiratory center. And while each particular location controls something different, collectively, they all control various aspects of pulmonary ventilation. So the respiratory center might make the difference between long, deep breaths and short, shallow breaths. And then finally, there's the midbrain, that green structure. In the midbrain, we see sites of integration or control centers for several visual reflexes. And let me introduce you to some of those visual reflexes. We'll talk about visual tracking, the pupillary reflex, and the accommodation reflex. And all of these are controlled by neurons in the midbrain. Visual tracking. So if right now, wherever you are, something were to fly across the room, you would reflexively follow it with your eyes. You would not only turn your eyes, but you would turn your head and your neck in order to track that moving object. That's a reflex. And I put a picture of Ms. Williams here because she is so amazing. And when I think of visual tracking, I think of people watching a tennis game as they watch that ball go back and forth from one side of the court to the other. Part of the visual tracking reflex is the movement of the eyeballs themselves inside the orbits. And to move your eyes, that requires stimulation of skeletal muscle. These are the extrinsic eye muscles. For each eye, there are 6 skeletal muscles that are attached to the eyeball. They're attached to the bones of the orbit. And when they contract, they pull the eyeball in different directions. So this is an example of what's generally considered to be an autonomic reflex, but involves the effector as skeletal muscle. So it's not always as clear cut as I have previously presented it. Another example would be, let's say, sneezing. Sneezing is generally regarded as an autonomic reflex, but notice that it does require some skeletal muscle in order to sneeze. Or to cough also involves some skeletal muscle. All right, the pupillary reflex, you can observe this right now. You can go and look in a mirror, and then if you change the lighting conditions, you will see the diameter of your pupil increase or decrease. In low light conditions, the diameter of the pupil automatically increases. It expands in order to allow more light in. And in high light conditions, such as standing in bright sunlight, the diameter of the pupil will constrict or narrow in order to let less light in. And the diameter of your pupil is controlled by a set of muscles, and these are smooth muscles. So the effector is smooth muscle, and the site of integration for the pupillary reflex is in the midbrain. And finally, the last visual reflex that we'll talk about is the accommodation reflex. Accommodation refers to your ability to focus on objects that are at different distances from your eye. So in a classroom setting, students might be looking up at me or looking up at the image projected onto the screen. You're looking at the slide and maybe that's 20 feet away. And then the student will look down at their notes, which is maybe 1.5 feet from their eye, and reflexively, the lens of the eye changes shape because different smooth muscles pull on it. So another set of smooth muscles in the iris of your eye are responsible for manipulating how round or how flat the lens of your eye is. And by manipulating the shape of that lens, you can change where that light gets focused on your retina. And this is called accommodation. All right, our next major brain region is the cerebellum. The cerebellum is located sort of between the occipital lobe, which is part of the cerebrum, and the brainstem. And the cerebellum does not initiate skeletal muscle movement, but it does play a huge role in smoothing and coordinating skeletal muscle movement. So your cerebellum has this incredibly high density of neurons. And I don't think that neuroscientists have a particularly thorough understanding of why the cerebellum has quite such a high density of neurons. That's still an area of investigation. We do know that the cerebellum receives sensory input from other parts of your brain as well as from the spinal cord. And the cerebellum is a massive seat of integration where the motor output arising will serve to coordinate and finely tune, finely smooth skeletal muscle movement. We do know that when somebody suffers, let's say, a stroke or trauma to the cerebellum, it can lead to an inability to execute fine movement. Like it might be very difficult for somebody to thread a needle, for example. They can also experience issues with staying still, finding their sense of equilibrium, their sense of balance can be disrupted. Their just normal resting posture can take on an odd appearance. And they have a more difficult time learning how to do motor tasks. So someone with an injury to the cerebellum might have difficulty learning how to bring a fork to their mouth or how to wash their face. In the lower right corner of this slide, there is a link to a video on YouTube. It's 1 minute long. That is also posted in Canvas under the suggested tutorial videos, and it's called Cerebellar Gait, and it's just a documentation of a particular patient who exhibits a characteristic way of walking after someone has sustained damage to their cerebellum, and that's usually a stroke or some type of head trauma. And when you watch the video, notice that the patient is not moving fluidly. They are not walking fluidly, but rather the patient walks sort of like a toddler who's learning how to walk. And so take a minute, pause this video, take a minute and go and watch that video to see how body movement is affected when the cerebellum is not functioning properly. This case astonished the world of neuroscience many years ago now. It's about 6 years old. This person was discovered when she complained of nausea and dizziness. She was having problems with her balance and she was upset to her stomach. A 24-year-old, I believe she lives in China, and she went to the physician with these complaints, and the physician ordered a CAT scan. And she, up to that point, she had been living a pretty typical life. She had a job and friends and etc. But when the physician saw the CAT scan, I'm sure his or her mouth dropped open. She does not have a cerebellum. Apparently, she was born without one of the 4 major brain regions and was living a relatively normal life. So she became the subject of a very interesting case study. And upon a very detailed neurological examination, it was discovered that she has difficulty pronouncing certain vowels, and that's a motor skill, right? Speech is a motor skill. Skeletal muscles are involved, and speech requires a great deal of coordination among many different muscles to produce speech. And it was also discovered that during her early childhood, she started to walk and speak later than the average, around age 6 and 7. But eventually she did learn to walk and talk, and everything was fine. And this is evidence of what is referred to as brain plasticity or neuroplasticity. And that refers to the ability of the nervous system to find a way to compensate for losses in function by developing new neuronal connections. And the brains of babies and young children are particularly plastic. They can overcome a lot. They can compensate to a great degree. And so her brain, this woman's brain, almost completely compensated for an entire region being absent. Our next major brain region is the diencephalon, and we'll talk about the thalamus and the hypothalamus. So the diencephalon is centrally located in the brain, and the thalamus is a major relay hub. Lots of different types of incoming sensory information comes into the thalamus and then gets passed along to one or more other brain regions for further processing. So let's look at the next slide. So we think that the thalamus itself can give rise to crude bodily sensations. So if the cerebrum, and that's our next major brain region, if the cerebrum was not working and incoming sensory input caused by something in your shoe passed through your thalamus, there is some evidence to suggest that that gives rise to what would be described as a crude or an unrefined sensation. So your thalamus could tell you, hey, there's this pain in my foot. There might be something in my shoe, but it wouldn't be able to distinguish between a pointy rock and a quarter. And you know those two things feel very different in your shoe. And you can easily tell the difference between a pointy rock in your shoe and a quarter, a coin in your shoe. So the thalamus will send that incoming sensory information onto other brain regions that then can perform a more refined processing. And next we'll talk about the cerebrum, specifically the primary somatosensory area, and we'll see that that's the area that allows you to really tell that there's a quarter or a pine cone or a pointy rock or maybe a piece of glass in your shoe. All right, so we're still in the diencephalon here. And the hypothalamus is also a major relay hub, and hypo means below, right? So the hypothalamus is inferior to the thalamus. And the hypothalamus is the site of integration for many, many autonomic reflexes, including, and we talked about on perhaps day 2, we talked about body temperature homeostasis. There are also clusters of cells here that are responsible for regulating your sense of hunger or satiety. What does it mean to be sated? Satiety refers to your sense of fullness, that you're finished eating. Your thirst center, your libido, is controlled by, or your sex drive are controlled by different clusters of neurons, all located within the hypothalamus. And your hypothalamus is physically connected to the pituitary gland. And another job of your hypothalamus is to control the secretion of hormones by the pituitary gland. Here's an interesting technique for understanding the functions of brain regions. So in this technique, we use model organisms. So you're looking at mice here because this would be unethical and impractical, excuse me, to perform in humans. And so scientists, in trying to understand the hypothalamus and specifically trying to understand the nervous system control of the sense of hunger versus the sense of satiety, they place electrodes into the brains of mice, and these electrodes allow the researchers to deliver electrical stimulation to very specific clusters of neurons in the brain. And so you can artificially induce action potentials in these neurons. You can cause them to light up and become active and see what the effects are on the behavior and the overall health of the animal. So when these electrodes are placed into the feeding center, which is one tiny region of the hypothalamus, and these electrodes are stimulated, it causes the mouse to overeat and to become fat. And when these electrodes are placed into a different tiny area of the hypothalamus called the satiety center, and these electrodes are turned on, this causes the mouse to undereat, and in fact, it will cause the mouse to starve to death. This image shows us the connection between the hypothalamus and the pituitary gland. That physical connection, that stalk, maybe you remember it from another course, it's not labeled here, but that's called the infundibulum. And the pituitary gland is called the master gland because it secretes so many different hormones, and many of those hormones in turn go on to control the secretion of hormones from other endocrine structures in the body. I'll show you an example in a moment, but first, while we're here, because it's so clearly displayed, the pituitary gland is two glands in one. There's the posterior pituitary and the anterior pituitary, and they function quite differently from each other. They each secrete their own sets of hormones. They are both controlled by the hypothalamus, but they are controlled by the hypothalamus in very different ways. In this image, we can get a sense of why the pituitary gland is referred to as the master gland. So up top in the anatomical diagram of the hypothalamus and the pituitary, you can see clearly the hypothalamus is physically connected to both halves of the pituitary, the posterior and the anterior. Those physical connections are what allow the hypothalamus to control the secretion of hormones from both the posterior and the anterior, although by very different mechanisms, which we'll talk more about when we get to the endocrine system. And then down below, those radiating arrows indicate that hormones secreted by the pituitary have an influence over a great number of tissues and organs in the body. And some of these organs are endocrine structures themselves. So notice the thyroid gland, which is around 7 o'clock, and notice the cortex of the adrenal gland, which is around 6 o'clock on this image. And hormones that are produced by the anterior pituitary go on to control hormones that are produced by both the thyroid and the adrenal cortex. So just to reinforce that the pituitary gland is the master gland because it regulates so many aspects. It regulates the activity of so many different tissues and organs. And we will talk a lot more about the pituitary gland when we get to the endocrine system. And once again, the pituitary gland is a great way to reinforce that the nervous system completely overlaps with the endocrine system in several locations in our body. In the pituitary gland, we have neurons that secrete hormones. The posterior pituitary releases neurohormones. And then Down below, we've already talked about the role of the adrenal medulla as part of the autonomic nervous system. It's like a modified ganglion, and those cells that produce epinephrine and norepinephrine are modified neurons. And technically, epinephrine and norepinephrine are neurohormones, although we typically just call them hormones. And our last major brain region is the cerebrum. The cerebrum is really what makes you, you. It gives rise to your sensory awareness. The fact that you even know that there's a rock in your shoe at all occurs because you have a cerebrum. It gives rise to your voluntary muscle responses. So if you're going to take off your shoe to take the rock out of your shoe, that is initiated in your cerebrum. Your ability to understand and use language, your ability to have abstract thoughts, like we can sit here and think about, well, what is love? And we can think about, well, what is truth and what is justice? Those are all abstract thoughts. Our creativity, our inventiveness, that's a hallmark of our species. We are inventive. All of those functions occur because we have these really large cerebrums relative to our body size and relative to other organisms. So the cerebrum is divided up into functional areas. What type of processing or what type of information is each region of the cerebrum in charge of? And so we have sensory areas, motor areas, and association areas. And I'll take you through specific examples of each. Each of these sensory, motor, and association areas of the cerebrum may be identified by a number. And these are called Brodmann areas. And Brodmann areas are named after Corbinian Brodmann, who was an early neurologist, a German. And a lot of early neuroscience came out of Germany in the early 20th century. And he published functional maps of the cerebrum that allow scientists to have a common language for talking about specific locations in the cerebrum. So neuroscientists might identify their work as pertaining to Brodmann area number 46, or Brodmann area number 7, or Brodmann area number 19. We will cover examples of each type of functional area in the cerebrum. Let me introduce you to their basic definitions. Sensory areas of the cerebrum deal with sensory input and allow us to have conscious awareness, conscious perception of that sensory input. Motor areas deal with generating voluntary body movement, and you also have conscious awareness over that. And then association areas deal with things that are neither sensory nor motor. So there are lots of functions in your cerebrum that don't involve any sensory input and they don't give rise to any motor output. So you can just sit back and contemplate the nature of reality, and that does not involve sensory input or motor output. You can experience emotions. You can recall memories. You can do a math problem in your head, and that doesn't require any sensory input or motor output, and those are called association areas. So, circled in green are the areas that we will cover as examples. Some of them are sensory, others are motor, and others are association areas. So the example sensory area we will cover is called the primary somatosensory area. And it occurs along a strip on the cerebrum called the post-central gyrus. So a major landmark in the cerebrum is called the central sulcus. And let me point to that. This is the central sulcus here. So a sulcus is a fissure. It's a crevice. And this is the central sulcus. And it goes from temporal lobe on the right side to the temporal lobe on the left side, with this structure here being the temporal lobe of the cerebrum. And so this central sulcus separates two gyri. A gyrus is a ridge, and the post-central gyrus that's highlighted in pink here, that post-central gyrus, because it's after the central sulcus, so we call it the post-central gyrus. And where after really means posterior. It's posterior to the central sulcus. That post-central gyrus is where we find the primary somatosensory area, and it incorporates Brodmann areas 1, 2, and 3. And let's think about what it does. Your primary somatosensory area is responsible for all of your conscious awareness of sensory input that's coming in through structures associated with your somatic nervous system. So your 5 senses, the touch receptors in your skin, for example, temperature receptors in your skin, vibration, etc. And in this primary somatosensory area, as we'll take a look at a picture of it in just a moment, there's a map of your entire body. There are different pieces of real estate, so to speak, so different clusters of neurons that are dedicated to giving rise to this perception, this conscious awareness of what's happening with this part of my body. So if you can feel the socks on your ankles right now, or maybe you can feel the sleeve of your shirt on your arm right now, that sensation is being generated by a particular place on your primary somatosensory area. And if we perform brain surgery while the patient is awake, they've been sedated and they've been given a local anesthetic, there are no pain receptors in the brain. So we can perform brain surgery with an awake patient. And if we electrically stimulate specific places on the primary somatosensory area, the patient will report, I feel a tingling in my elbow, or now my big toe itches. They'll report perceiving sensations from a very specific part of their body. So this is your primary somatosensory area drawn out to reflect and I always refer to them as areas of real estate, with these sections that contain neurons that are dedicated to perception over very specific parts of your body. And here, the body parts are drawn to reflect the amount of real estate, the amount of area of the primary somatosensory area dedicated to perceiving sensations arising from that particular body part. So for example, look at the back of the knee. Look at the back of the knee, sort of on the top right of this diagram, and then compare the size of the back of the knee to the size of the palm of the hand. And the back of your knee is, maybe the palm of your hand is a little bit bigger than the back of your knee. They're roughly the same size, but you have far greater sensory awareness in the palm of your hand relative to the back of your knee. So you'll be able to detect sensations with a much finer degree of granularity, with much greater resolution when that stimulation is applied to the palm of your hand versus the back of your knee. This is a different artistic representation of similar concepts. So on the left, where we're looking at those 2 cerebral hemispheres, that brown colored post-central gyrus is the primary somatosensory cortex, and then it's blown up from there if we just go a little bit to the right. So in the middle of this image, the primary somatosensory cortex has been expanded and color-coded, where each color corresponds to a different body part, and it's connected to your somatic nervous system. It's all part of your somatic nervous system. And then if we look all the way to the right with that very funny image, that is a human body, obviously drawn in proportion to reflect the proportion of the primary somatosensory cortex dedicated to perceiving sensations arising from various parts of our body. And so look at the back of the knee. It's tiny relative to the palm of the hand. So you can clearly see what the most sensitive parts of our body, they are our hands and feet, they are our mouth, our lips, our faces. And think about over the course of human evolutionary history, what do these body parts do? Your hands and your feet are in constant, this is before shoes. So yes, your hands and your feet are in constant contact with your environment as you walk and as you manipulate objects. And then of course your face, your lips, your tongue, that's for communication. Humans have expressive faces and have the most expressive language known. All right, our example motor area, or one of them, is the primary motor area. And if you look at the image, the primary motor area is on the precentral gyrus in the cerebrum. So we were just talking about the primary somatosensory area, that's highlighted in blue. The primary motor area is highlighted in red, and the fissure, the deep crevice between the two, is called the central sulcus. On the primary motor area, there is also a map of your body, and your voluntary bodily movements arise from these regions of your primary motor area. If we do brain surgery on an awake, aware, behaving patient and we stimulate particular regions of the primary motor area, we can cause body parts to move. So if an electrode is placed on the part of the primary motor area controlling the triceps, for example, the triceps are the muscles in your upper arm, on the back of your upper arm. If we apply electrodes to that area of the primary motor area, we can cause the patient's arm to extend at the elbow. They'll unbend their arm just by placing electrodes at that particular brain region. Similar to the primary somatosensory area, there is a map of the body also present on the primary motor area. And so if during that brain surgery we wanted to get the patient to extend their arm at the elbow, we might place an electrode right around here and stimulate these cells, and the triceps would contract to unbend the arm. This is a Far Side cartoon by cartoonist Gary Larson. And Gary Larson has a real biological bent to him, and he became a very successful cartoonist. And there's always a nugget of truth in all of his cartoons, and obviously he was very popular among the sciencey crowd. And so here are a bunch of neurosurgeons doing brain surgery, and one of them says, "Whoa, that was a good one. Try it, Hobbs. Just poke his brain right where my finger is." And then we see the patient's leg extending at the knee, and maybe there's a little hip flexor action as well. We're going to cover an example of another motor area, and this is called Broca's speech area. So I'll remind you that motor areas are responsible for giving rise to motor output that stimulates skeletal muscle. So this is called Broca's speech area. So this area is probably involved in causing muscle contraction required for speech. And that is true. And Broca's speech area includes Brodmann areas 44 and 45. By the way, please don't memorize any Brodmann areas. Not necessary. We can always look those up. So let me introduce you to another early neuroscientist. He was French, and Paul Broca did amazing work, made many discoveries by working with patients who have aphasia. And there are many different, now we know that there are many different types of aphasia. But generally speaking, aphasia is the inability to comprehend or to use language. And Paul Broca was very meticulous in his research. He would spend days and hours and days and weeks with individual patients, making very detailed notes about all of their capacities and all of their incapacities. What could patient X do and what could patient X not do in excruciating detail. And even today, we still use the preserved brains and the preserved notes made by Paul Broca to refine our understanding of neuroanatomy. As our microscopy techniques have gotten better, we can still use those preserved brains to see where the damage occurred in one of Paul Broca's aphasia patients and look at his notes to see how that specific damage that we now can understand quite well with the microscopy techniques that have developed since his death, and we can look at his notes and we can match function with form. We see the damage and we see how function was disrupted or how function was not disrupted based on his detailed notes. So there are many forms of aphasia. Aphasia can show up as difficulty speaking, difficulty understanding, comprehending language. Maybe the person is no longer able to make sense out of written words, and maybe the person is no longer able to write. So we'll talk about Broca's aphasia. We'll also talk down the road, we'll also talk about a different type of aphasia called Wernicke's aphasia. So Broca, let's start with that. Broca's aphasia is caused by damage to Broca's speech area, likely caused by a stroke. So there was this lack of blood flow to that particular region of the brain, and then the patient has a particular set of symptoms regarding their speech. Their ability to talk is slowed down. Their language could be slurred. They might have difficulty forming the words clearly, or they may lose their ability to speak entirely. It takes, if they can speak, it takes great effort to produce speech. And so what I would like you to do is to watch this YouTube video. And I've added this slide to this lecture recording. In a regular classroom setting, we watch this video and we talk about these questions. And so I would like you to also pause this video, go and watch this interview with Randy. Randy has Broca's aphasia, which is also known as expressive aphasia. And here are 4 questions to consider. I will also ask you to consider these 4 questions down the road when we look at a different patient who has a different type of aphasia. So as you watch this video, ask yourself, can you tell, do you think that Randy knows what he wants to say? Do you think that he has an awareness of how he sounds? Does he know what's happening? Do you think he seems frustrated? Can you understand his story? And can you paraphrase it? Like, what is Randy trying to say? Can you tell his story in a few sentences? And finally, imagine that Randy is speaking a language that you don't understand. Just pretend the whole interview is in Turkish or Portuguese or Tamil. If you were watching, would you notice that there's a problem, or does everything look okay? So keep those things in mind. Watch this video, come back, and let's talk about these questions. Okay, do you think he knows what he wants to say? Yes, it's very clear that he knows what he wants to say. Do you think he knows how he sounds? Yes, and yes, he seems frustrated. Were you able to understand and could you paraphrase his story? Yes, his friend picked him up. They went to go and get something to eat at McDonald's. They went to go see the James Bond movie. It was a great day. And then imagine he was speaking another language, one that you didn't understand, and you watched this interview. Would you notice that something seemed off or wrong? Maybe, maybe not. He doesn't seem happy, and he does seem frustrated. But it's sort of hard to say that something is clearly off. Oh, that guy from The Big Bang Theory. Are you Broca's aphasia? Because you leave me speechless. All right, so Paul Broca's work led to an understanding of what's now called hemispheric lateralization. And that refers to there's an asymmetry in the cerebral hemisphere. So you have a right cerebral hemisphere and a left cerebral hemisphere, and they do not mirror each other in terms of functional areas. So for most people, Broca's area occurs in their left cerebral hemisphere. For some people, it's in their right, but the vast majority of people have it in their left. And for most of us, we will never know. We will live our good, rich, healthy lives until they're over, and there is no reason for us to know if our Broca's area is in the left side or the right side of our cerebrum. So there is this asymmetry, this hemispheric lateralization. So certain functions tend to occur in one side versus the other. And this is super duper generalized. There is quite a bit of individual variation. So no two people's cerebrums are alike. But in the left hemisphere, most of us tend to have our centers for language, for doing math, for engaging in logical reasoning. If A equals B and B equals C, then C equals A, right? That's logical reasoning. And then in our right hemisphere, most of us have centers that deal with spatial reasoning, like you might use if you're playing the video game Tetris. Face recognition. There's a face center in your brain that allows you to look at a particular face and recognize it as someone you know or someone you don't know. And then our ability to generate and appreciate music also occurs in the right hemisphere for most people. So I mentioned what Paul Broca was doing, and he engaged in a very common neurological technique now when we can use patients who have a neurological deficit, so living, breathing humans who have suffered some type of stroke or head trauma, or maybe they were just born without a cerebellum. We can study patients and their neurological deficits to understand the role that a particular brain region plays in the normal, healthy, typical body. All right, so I gave an example of a sensory area, 2 examples of motor areas, and here's an example of an association area. And I'll remind you, association areas are responsible for neither sensory input nor motor output. And our example is Wernicke's speech area, Brodmann area number 22. So Wernicke's speech area is not responsible for producing speech. It doesn't generate the motor output to the muscles of the throat and the face. Rather, Wernicke's speech area is responsible for your ability to comprehend language. You hear a word like dog, and that conjures an image or the idea of what a dog is. If Wernicke's speech area was not functioning properly, you would hear the sound dog and it would not mean anything to you. Just like if I go bleh, that doesn't mean anything to you. So Karl Wernicke, also an early neuroscientist, also German, also worked with aphasia patients. And so he sort of was picking up where Paul Broca left off in some respects. And he noticed that there were different types of aphasia, and not all aphasias were caused by damage to what we now call Broca's area. But damage to other regions of the brain can also lead to different types of aphasia. And now we have a type of aphasia called Wernicke's aphasia, also known as fluent aphasia. So people who have fluent aphasia or Wernicke's aphasia, they are able to generate language, no problem. They can speak. They just can't speak clearly. The muscles are all working. Their language is clear. The words are very fluid and very easy to understand. They just don't make any sense. They appear to have no understanding of what the words actually mean. And sometimes it's described as speaking in word salad, like a tossed salad. The words are just randomly thrown together without any context or meaning. So what I'd like you to do is pause this video, and please go and watch this YouTube video. And you'll see a brief interview with Byron, and Byron has fluent aphasia, also known as Wernicke's aphasia. And here are those same 5 questions that you thought about when you watched the interview with Randy. So pause this video, watch Byron, and come back to this video, and then let's talk about these 5 questions. Do you think Byron knows what he wants to say? It's hard to tell. It's really hard to tell. Do you think he's aware of how he sounds? No, he seems completely oblivious to the fact that there is nonsense coming out of his mouth. Does he seem frustrated? Not at all. He seems like a pretty happy guy having a normal conversation. Can you understand and paraphrase his story? Not at all. It's not at all clear what Byron wants to or is trying to say. And then imagine he's speaking a language that you don't understand and you're just listening in on the conversation. Would you notice a problem? Not at all. Notice that his rhythm, his intonation, his facial expressions all appear perfectly normal. He's retained the typical cadence of language, but just the words are strung together in the wrong order such that they don't make any sense. And notice that it's also not clear that he understands the questions that he's being asked. All right, so in order to have a conversation, you require many different regions of your cerebrum. And just a very simple pathway is illustrated here. So let's make up that this is a mom and her son. And she's saying, hey, thanks for doing the dishes yesterday. And in order for him to hear that, those sound vibrations, those vibrating air molecules, trigger action potentials in the cochlea, which is in the inner ear. It's a sense organ. And those sound vibrations are translated into electrical signals, into action potentials in the cochlea. And those action potentials travel along a cranial nerve, the vestibulocochlear nerve, and enter the brain. And that incoming sensory information passes through the thalamus and is directed out toward several places, one of them being the primary auditory area, not shown here, but it's in the cerebrum. So now the son knows that he's hearing his mom's voice. And in Wernicke's area will allow him to make sense of her language. Oh, she just thanked me for doing the dishes yesterday. And now he wants to say, well, you're welcome, Mom. It was my pleasure. And so from Wernicke's area, information gets sent to Broca's speech area where he, in Wernicke's area, he can think of the words that he wants to use. Well, thanks, Mom. It was my pleasure. And in Broca's area, the motor output is initiated. That motor output that is needed to stimulate the muscles of his face and throat is initiated and then sent to the primary motor area. And then that primary motor area will send the motor output out of the central nervous system along somatic motor neurons to the skeletal muscles required for producing those words. You're welcome, Mom. It was my pleasure. Here's a different diagram essentially illustrating the same flow of information. During a conversation, words are processed by the primary auditory area, and that language gets translated in Wernicke's area, meaning the language can only have meaning if Wernicke's area is working properly. And the response, you're welcome, Mom, it was my pleasure. That response, those words, are also put together conceptually in Wernicke's area, and Wernicke's area then communicates with Broca's area, which gives rise to the initial motor output, and that information travels to the primary motor area. And the motor output that will leave the central nervous system arises in that primary motor area and travels along motor neurons out through cranial nerves to motor neurons that control the muscles required to form those words. You're welcome, Mom. It was my pleasure. All right, and that brings us to the end of segment 4 of 5 on the nervous system. And in today's video, we talked about functions of the spinal cord, number 4, and we talked about number 5, select functions of major brain regions. And in segment 5, we'll pick up with neuroplasticity. See you there.